Okay, I know I said I was gonna finish it a couple hours ago, but I just doing something for my colleague. So still got so many hours until the end of the day. A-level biology papers, A-level papers, processes 2023. Open that. I'm gonna use my brain more. I went to read a book, then I put the book down. I decided I don't want to read the book anymore. I want to read a thriller. I want to read something that gets my blood going. I don't want to read some flipping bumbleclot thing about the brain. Okay. Let's make this. Okay. Where's the thingamabob? Where's the thing to type? Where is it? It's just gone. See, it should just disappeared. Do I have to go out of full screen? Okay, so in the dark, so light is controlled and is an independent variable. So the direction of shoots and routes are not affected by direction of light / phototropism. So most likely this is going to be along the same thing. So I'm now going to say placed vertically to avoid effect of geotropism for fair measurement. I don't actually know why. I don't understand. Where's my typing thing gone? It just won't let me type. Hold on. I don't know my mic. Is my mic working? Okay, the mic is working. We're gonna open it again. It's just not here anymore. Okay, it is a control to see / check if waltzing concentration actually has an effect. I think that's it. I can't. It is a control to see if it has an effect. Get the calculator out. It's 19%. I'm going to put a plus there. Oh, this is going to be like a stupid. Oh, what? Okay. I'll just explain what I would do. Concentration would be here to here, and each dot would be an increase in fold. And I think I'd have to do something like this. Do negative 100, positive 100. Yeah, I think I'll have to do something like that. And do one for shoot, do one for root. That's what I would do. Okay, I'd create a serial dilution, but we want to. What's the things? Concentration. Oh, so this is 1 times 10 to 2 is just 100, right? Yeah, it's just 100. So you just dilute it by a factor of 10. Dilute by a factor of 10. So remove 1 centimeter cube from the original and add 9 parts distilled water. Dry pipette each time. I say bottle of meniscus, that's not a precaution though. I have no clue. I actually don't. This beaker is dry slash measuring cylinder. I can't think of anything else. Decreasing slash increasing by a factor of 10. That's the only way I think I could describe it. I do maths and I couldn't even. Do not allow trophism. What? Allow for geotrophic. Oh, tropic. Yeah. So I said they are geotropic. I think I'd get the mark for that. It says allow in dark to prevent phototropism. Yeah, so it's vertically to avoid effect. I said avoid the effect. No. Give myself 1 mark for the phototropism, but I should have said, yeah. Acts as a control. Compare. I'll give that, but I really should have said the word compare there. Plus 19%. Get to 2 sig figures. Let's see, what do they want on this graph? Whoops. Okay, I think I would have got 4 marks on that. That looks all solid. Should get a, flipping hell, man, I keep forgetting. Should get a mark. So I should have said mixing each time. See, I should have thought, okay, concentrate. Yeah, I should have thought of that. That's so silly. You're decreasing by a factor of 10. Yeah, I had no clue what else to say to that, but I got the mark. Do not change by time. I guess what? 1, 2, 4, 8, 10, 12, 13. Let me measure the other one as well. 3, 6, 7, 12, 14, 15, 18. Okay, I actually saw this. I accidentally skipped down on the mark scheme, but I knew what it is anyway. What would you say, synapse? I would say synapse. No, I would say synaptic cleft. first neuron in the pathway. A. Okay, so if we look here, how do neurons send pathways? They produce acetylcholine in vesicles. These vesicles move to the membrane. Membrane opens up. Vesicles poof out. That's what's happening here. It's just not happening here. The acetylcholine then binds to the thing. Tiny sodium ion channels open and sodium floods in. So it's flooding in here because there's no things. So yeah. Ion channels. This is an example of positive feedback because when it opens, negative 55 millivolts is the threshold value. Once that happens, more and more come in. Depolarization. Movement. Notice how it doesn't say human side scan. This is any side scan. Movement. Meiosis. Well, I'm thinking of spindle fiber. I'll say intracellular transport. Structural integrity and maintain shape of the cell. I know it holds the nucleus in place. Motor proteins can transport. Suggest how mutation genes could cause a disease in the nervous system. Okay, nervous system. Action potentials propagating. Okay, so I'm thinking, yeah. I just cut my lip open. Nervous system is all about action potentials propagating, right? So we need to think, what could stop this? Okay, we just did earlier vesicles. So vesicles are no longer able to move along a synapse or a cell / neuron. Acetyl exocyte host. So no release of neurotransmitters. No binding to postsynaptic neuron. Propagated. You see where I could have got this first thing wrong, but if there's a different, the nervous system is literally just those things being our disease, cognitive function. I'm not gonna go bother explaining that because it's all kind of self-explanatory. Like you have to type like the person doesn't know what they're reading. yeah, so I was synaptic cleft. I was actually going to put synapse approved, but I thought I wouldn't get the mark, and I was right. Vesicles, yeah, the exact 1 mark. That's a bit much. Positive depolarization. Okay, so I got movement of cell. Literally bang on. What the hell? Strengthening / supporting cells. Movement of named. Oh, movement of named organelles. Allow track for motor proteins. So literally got that right. Hold. I wrote holds nucleus in place. So I'm actually going to get Gemini up here. Hello, Archie. I need to bring up Mohammed's chat. Because, yeah. Okay, I'm not going to bother logging it to work out. Okay. Hold organelles in place. Huh. So you could have literally gotten 3 marks from just mentioning the flipping mitosis. Just saying it forms a spindle. Movement of chromosomes. Why? What's the point just signing? There's just no point in signing me out as I mean, I've lost my chat. I said nucleus, I was right. But I doubted myself. But yeah. Okay, we're gonna see if I was right about the mark scheme for this one. Why is it not? Get a new keyboard. Anyone want to get me a new keyboard? I'll do like free tutoring, something like that. Come on. Bro. Oh my days. Okay, let's hold on. It's change in structure of the protein. I didn't mention that, but I said no movement of vesicles. I said that. So I said less synaptic transmission. Action potentials do not continue from one to the next. Boom. I literally got the mark. I said the first point could be wrong. Could change diameter of axon. Effect. Like speed. do that, it could affect one-side integrity. Yeah, okay. Literally knew what I was talking about. I just spoke about what damage to a nervous system, what technically would damage be. Or, yeah, anyway. 2, 5, 7, 8, 9. Just got fucking 9 out of 9, mate. 9 out of 9, mate. Come on. 9 out of 9. Let's check the mark scheme. Is that 9? Oh, it doesn't tell me. Oh, it's 9 out of 9. Anyway. I think I've only lost like 3 marks so far, and I'm nearly done with the paper. So yeah, you know, that's kind of good. Okay, it just doesn't let me type anymore. That's fine. It's true. True. The skin sweats, I suppose. It excretes some metabolic waste products because it smells, so it's probably bad stuff. Carbon dioxide is formed by dealimation. I think that's true. I think it's true. I know it's producing the ornithine cycle, which is the process of dealimation. False. It's more soluble. Hence why we make it. Yes, that's true. Oh my days! Got the flipping most bookiest thing. Why is that just happened? Okay, label the parts. Okay, medulla, pelvis, cortex. T is medulla. U is cortex. Okay, this is the Bowman's capsule. You should just know when you see a thick thing, it says, "Can you?" That's Bowman's capsule. That's glomerulus. The filtration definitely doesn't happen in the pelvis. It just looks like it happens here. Yeah, what happened in you? When it goes down deep in the medulla, that's like the loop of Henley. So it might actually happen in you. You know. But yeah, that's where blood is first. I think it might be you. Because when it goes down the Bowman's capsule, no, when it goes down the loop of Henley, so it goes deep in the medulla. So I reckon it's happening in you. Yeah, see? I'm sorry, 1 kilogram. It's just what happened to the 1 kilogram. Maybe I cut the hair. Okay, if I link this to biology, I'm gonna say that maybe it's like grams of glucose. So I'm gonna say like glycogen stores depleted. Used up. Fat stores depleted slash used up. Aerobic. Used for respiration. Used in for respiration of mitochondria to produce energy. Snipped off by enzymes. Converted into CO2 slash ATP plus water. I can't really think of anything else to add to that, to be fair. Okay, I think I've actually got a 19A wrong. Okay, this thing just doesn't want to work, does it? It just doesn't want to work. That's fine. I don't want it to work either. Why is this oh, it's actually right. It's all right. Oh, it's actually used in. Okay. Medulla cortex pelvis. Allow renal pyramid. Okay, it's weird. It's not pyramid. U. CW. I don't think. Well, I put X, no? Actually, new. I've actually made like 10 flashcards on the. Maybe I should explain my thought process more so I stop making these stupid mistakes. Sending limb is impermeable water. I literally was going to write that and I said, know what? Doesn't matter. Doesn't matter. Okay, I got that. Active transport, that. Diffusion, I got that. Okay, so I got one. Okay, I got 4 marks. I just got 3 marks. I got 4 marks. I got 4 points, but I got full marks and it was 3. So you're going to mention, because... What? So I don't know why I'm so fucking inspired by that. Body fat slash glycogen broken down, used for respiration. Used in respiration, got that. Produce carbon dioxide and water. I was like, this is just naming just random things now, but I actually got the full marks. Now, I was even going to say it's excreted by the lungs. I was like, nah, I don't need to do that. But it would have been a mark in case I didn't get the others. I lost 1 mark there. 2, 5, 7, 9, 12, 14. 2, 5, 7, 9, 12. I wonder if she stopped responding to me because I sent a photo of those paintings and they're all girls. That's probably why. Why did you just message me like this? Hello. Defines an endotherm. Keeps a stable internal environment. Into outside environmental changes. Advantages: lower metabolic rate, disadvantage: at mercy of the environment. In winter, processes slow down, maybe forced into hibernation, less time to gather resources and mate. I'll just say that. Lower metabolic rate, less reliant on food / eating. I just spilled water everywhere. I did. Six markers. Okay. Now, ectotherms. Ectotherms do not regulate their internal body temperature with metabolic processes. Whereas endotherms do, such as human mammals can produce sweat to lower body temperature or start shivering to warm the body up. Endotherms do not sorry. Ectotherms do not have these processes, such as lizards. I don't know. Ectotherms do control body temp with behavioral such as lizards moving on to dragons. Endotherms, however, do use behavioral responses as well as humans slash dogs don't swear. What do dogs shiver? Humans into shade or sunny or high sunlight spots due to preferred due to overheating slash being cold. Okay. So first statement is correct in terms of it's right for the behavioral for ectotherms, but wrong for endotherms. And I'm just gonna add endotherms control the internal body temperature with metabolic processes. I don't think there's anything else we need to add to that. Flipping out. I only got like 2 questions there. Infection by pathogens as bacterial environment often cause release of stimulate the enzyme COX-2. Increase in... Okay, so it says hypothalamus and it says thermogenic. Hypothalamus is where the heat loss and heat gain center is. So thermogenic set point, I'm guessing, is heat. Heat set point is the baseline heat. Increase in the internal body temperature. I don't know. pain center. This is infection by pathogens. So faster metabolic rate / work of the immune system to fight pathogens. PG2 centers are located on the cell surface membrane. State 2 functions of the cell surface membrane. If I illustrate body action. So PG2, so cell signaling. So PGE is just a functions. Everything I got is cell signaling. I'm going to say cell compartmentalization. Oh, I should have said uses metabolic processes. Yeah, so I said lower metabolic food, less reliance on food, more energy, less time spent feeding source food, slows, less active at lower temperatures, hunt, find food. Huh? Yeah, I'll give myself a 5. If I writ that out by hand, I would have filled out the entire thing and it would have covered most of those points. I don't increase hepatic will result in fever. To help kill, prevent, increase. Yeah. Causes increase in higher body temperature. I hate how it says will result in fever. Well, let me ask Gemini. Gemini, my G. Whoops. Wrong screen. What would you mark this? Watch it say 0 out of 2, and then I'm just never listening. say here? I should have said acts as a barrier. That's all like translation stream. That's just that. Seal the borehole with tape. To block the tree. Stand and die leaking and staying within the system. Okay. So they want to see how it moves up and down the stem. If you want to view this in a laboratory, I'm not going to mention anything like nuclear magnetic resonance imaging. You just want to cut the stems. Cut the stems longitudinally. And then that's all I could really think. You could just watch it. So I guess you could say cover slip over stems and view under a microscope. So I could really think I could add to that. It's not really much you can add to. I suppose you could say, no, you can't really. Plasma. Okay. Of greatly increased mineral concentration of the soil. Increasing. Basically, right, I'm not going to explain my points because I'm just going to think. I don't actually know what they're asking. So I'm just going to write down what's happened in scientific terms. This increase in the solute concentration dramatically. ions are no longer pumped out of root hair cells against concentration gradient, water no longer moves down the water potential gradient into the root, but stay in the soil. Plants then have decreased water supply / resource, cannot undergo photosynthesis, light dependent photosynthesis, no assimilation of proteins / lipids to grow and develop. Okay, essentially the same effect we seen in the plants and water solution contains cyanide ions that inhibit ATP synthase. Yes. As the production of ATP will be halted / stopped, the enzyme no longer produce ATP synthesis and respiration would stop. So what do I even say to this? So no energy is no ATP is made for the cell to carry out metabolic processes. And I just realized this is linking it how it's not able. So root hair cells cannot actively transport these ions against the gradient. Actually, wait, no. If they want to draw water in, they'd actively transport them in. There is more light dependent photosynthesis occurring, which requires the photolysis of water in photosystem 2 to accept these electrons, which are excited from the higher light intensity. That's my only thing. I would say to cool down, but that sounds really fucking stupid. Like plants. Actually, no, because it says temperature's the same. That actually could have been a thing. Maybe he said that so students don't think that. Who knows? Movement of water from root to leaves. all my days. I don't actually know it was that easy. To prevent air break-in, to reduce evaporation, prevent entry. I forgot about those things. How was that? Did I say low? Actually, I'd give myself 1 mark there, I'm being real. Eight, 19, 33, 40, 42, 55, 63, 73, 77, 83. Can I pretend we didn't see those search history? Yeah, my great boundaries. brother, this better not be my Wi-Fi. It ain't even my Wi-Fi, it's my nan's Wi-Fi. Realistically. Processes 68. You do 83. 83. Take away 68. Got 50 marks above an A star. Okay. Let me go on my Google Sheets. Oh no, the Diddy sheet. What's wrong with me? Loves messaging me. That looks like an email. It looks like an important email. So, what did I say? A star 15 plus. I'm gonna go with this color scheme because I prefer purple. Yeah, like purple is a good color actually. Like this purple, yeah. That's like a Steve Harvey kind of nigger purple. Anyway, my heart's going because this could have been an important email. Oh my fucking god, man! It's just Siren Sets to College by Unsignable. I'm not signing. I might just actually block this. It looked like a really long, lengthy email. So, oh my lord, is this a uni offer or something? Fucking no. Leave page. Anyway, I said I was gonna do this thing. Okay, a student completes a series of assignments. Let's get oh. actually am just gonna ask AI this question, because I don't actually know. Okay, so we've actually done this. 83 is an A star. I think surprise. That's based on OCR-A. And I'll ask ChatGPT in the meantime.